Welcome back to another Think Spree video, where curiosity meets clarity and learning doesn't come with homework. Today, we're diving into something that sounds like a breakfast cereal but could literally rewrite the future of humanity. That's right, we're talking about CRISPR. Not crispy. Not CRISPR, like your fridge drawer. CRISPR. The gene editing tool that's changing everything we know about biology, medicine, and maybe even ethics. Now, before you go imagining mad scientists in lab coats turning house cats into dragons, take a deep breath. It's not quite that sci-fi. But it is powerful. And a little wild. And by the end of this video, you'll know exactly why. Oh, and while you're here, don't forget to hit that subscribe button. Think of it like editing your YouTube feed to be 100% smarter. CRISPR won't change your genes, but subscribing might change your algorithm. Let's get into it. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, it's a mouthful. But don't worry, we're not going to throw a textbook at you. Here's the short version. CRISPR is a natural system that bacteria use to defend themselves against viruses. Scientists figured out how to turn this bacterial defense system into a precise gene editing tool for other organisms. Including, potentially, humans. Let's rewind for a second. When viruses attack bacteria, they inject their DNA inside them. Over time, bacteria developed a clever little defense mechanism. They'd snip out pieces of that viral DNA and store it in their own genome. Kind of like taking a mugshot. If the virus came back for round 2, the bacteria would recognize the DNA, use an enzyme called Cas9 to chop it up, and boom, no more virus. This system, CRISPR-Cas9, was discovered by scientists in the early 2000s. And in 2012, two researchers, Jennifer Doudna and Emmanuelle Charpentier, figured out how to turn it into a tool to edit genes in any living thing. Not just bacteria. Plants. Animals. People. And for that, they won the Nobel Prize in Chemistry in 2020. Casual. So what makes CRISPR such a big deal? Well, in the past, gene editing was like trying to do surgery with a chainsaw. Slow, messy, and extremely risky. CRISPR is like upgrading to a laser scalpel. It can find a specific sequence of DNA, cut it out, and replace it with something else. Quickly. Cheaply. Precisely. You want to fix a gene that causes disease? CRISPR can do that. Want to make a tomato plant that's more resistant to drought? CRISPR's your guy. Want to make a mosquito that can't carry malaria? CRISPR has entered the chat. And here's where things start to get a little spicy. Because editing genes means you can fix inherited diseases. But it also means you could enhance traits. Strength. Intelligence. Eye color. You know, the usual sci-fi territory. And that opens up an ethical Pandora's box the size of a small moon. Let's talk about some real-world examples. In 2017, scientists used CRISPR to remove a gene that causes a deadly heart condition called hypertrophic cardiomyopathy from human embryos. Those embryos weren't brought to term, but it showed that CRISPR could potentially prevent certain genetic diseases before a baby is even born. In agriculture, CRISPR is being used to create crops that resist pests without needing chemical pesticides. That could reduce environmental damage and boost food security. Imagine strawberries that don't spoil in 2 days. Or wheat that can grow in salty soil. Or corn that tastes like cotton candy. Okay, that last one might be a little ambitious, but the point is, the possibilities are huge. Then there's medicine. Researchers are already testing CRISPR in clinical trials for things like sickle cell anemia, certain cancers, and even HIV. The idea is simple but powerful: find the faulty gene, fix it at the source. That's not a treatment. That's a cure. Now, let's address the glowing elephant in the room: designer babies. Yeah, we went there. In 2018, a Chinese scientist named He Jiankui claimed he had used CRISPR to edit the genomes of twin baby girls to make them resistant to HIV. the scientific community did not respond with applause. He was condemned globally, arrested, and sentenced to prison. Why? Because editing embryos that will grow into humans is a huge ethical leap. And no one knew if it was safe. Or even effective. Editing one person's genes is one thing. Editing the genes of someone whose changes will be passed down for generations? That's a whole other level. What if there are unintended effects? What if you cause more problems than you solve? And let's not even get started on the potential for inequality, where only the rich can afford to make their kids taller, smarter, or stronger. You don't need a PhD to know that's a bad recipe. So where does that leave us? Right now, CRISPR is like a superpower we've just discovered. And like all superpowers, it comes with responsibility. Scientists are moving carefully, and there are strict regulations around what you can and can't do. But the future is closer than most people think. In the next decade, we could see treatments that permanently cure genetic disorders. We could see gene-edited animals that are more sustainable for farming. We might even use CRISPR to bring back extinct species, Jurassic Park style. Though, for the record, maybe don't start with the T-Rex. Let's try a dodo or something less bitey. There's also talk of using CRISPR to fight climate change. No, it won't turn off fossil fuels, but it could help engineer trees that absorb more carbon, or microbes that break down pollution faster. It's like giving nature a boost with its own building blocks. And this is where your brain should be doing cartwheels, because the more you think about it, the more you realize CRISPR isn't just about fixing what's broken. It's about redesigning life itself. But that's a slippery slope if we're not careful. So what's the big takeaway? CRISPR is one of the most exciting, promising, and controversial breakthroughs of our time. It's not magic. It's not science fiction. But it is a tool, a powerful one, that could change how we treat disease, grow food, protect the planet, and maybe even understand what it means to be human. It's also not perfect. Off-target effects, ethical concerns, long-term risks, all of that is still being studied. But if history has taught us anything, it's that when humanity gets its hands on a tool this powerful, we have to choose carefully how we use it. Because once you edit the script of life, there's no undo button. Now, if this video made you a little smarter, a little more curious, or just gave you an excuse to delay your chores, do us a favor. Hit that subscribe button like you're correcting a genetic mutation. Tap the bell so you don't miss future ThinkSpree uploads, and give this video a like so we can keep bringing you content that hits harder than a Cas9 enzyme. Thanks for joining us on this gene splicing journey. We'll catch you next time, right here on ThinkSpree. Until then, stay curious, stay cautious, and maybe double-check what's in your fridge CRISPR drawer, just in case.